Okay, this presentation is an overview of 2.2.3 biochemistry. So we're going to be focusing on macromolecules. So first we'll talk about the four major macromolecules. So the first one is carbohydrates. Carbohydrates are also known as sugars. So some sources of this would be bread, pasta, soda, candy, things like that. So first, let's just take a look at the structure of a carbohydrate. So what you're going to be noticing is this ring structure. We have a 6-carbon ring typically. It could also be 5 as well. And we actually shorthand these molecules because it would take a long time to draw them if we included all of the components. But basically, each point here would represent a carbon. So instead of drawing in all the carbon and hydrogen, we can just shorthand it. When you take an organic chemistry class, you will learn all about that. So for right now, we'll just focus on how we normally represent those sugar molecules. Okay, so then if we have a disaccharide, di means 2. So we have 2 sugar molecules put together. So here's 1, 2 sugar molecules. Then if we get more than 2 sugar molecules put together, we would have a poly meaning many, saccharide. So see here we have 1, 2, 3, 4 different sugar molecules put together. So we have repeating subunits. Okay, so they should all look like the monosaccharide that we have above. Okay, right here. So if we put a bunch of those together, we'll have a polysaccharide, which could also be known as starch. Okay, so that's important that you understand that starch and polysaccharide are pretty much the same thing. All right, so again, monosaccharides, mono means 1, so it's talking about 1 sugar. Some examples of that would be glucose, fructose, galactose. So glucose is the one we've been talking about in terms of diabetes. You may have heard of high fructose corn syrup, and that's one of our sources in our diet of fructose. But also if we have glucose and fructose put together, that's sucrose. Okay, so this molecule right here, fructose, and we have our glucose. So this is the simplest subunit, so that's why it's called our monomer. Okay, and then when we put them together, we can get a disaccharide, meaning 2 sugars. So examples of that would be sucrose, which is just table sugar. Lactose is the sugar in milk, so if you have issues digesting milk, you may lack the enzyme that breaks down lactose. And these can be broken back down into the monosaccharides. Okay, and then polysaccharides, again, poly meaning many. Examples of this would be starch, glycogen, which was the sugar that we store in our cells. Fiber is also considered a polysaccharide. And again, we can break these down into monosaccharides. So we'll address how we break those molecules down or form them in a future slide. Okay, so another macromolecule that we are concerned with is protein. And protein is made up of amino acids. These are very, very complex molecules, typically very large. They could be thousands and thousands of amino acids long. So we have a lot of different models that will represent them. But based on those amino acids, you're going to get a different shape for the protein, and we'll discuss that later in Unit 3 when we're talking about sickle cell. And it's actually very simple to recognize amino acids. Amino acids are always going to have this amino group. So you'll see nitrogen. Switch colors. Okay. So you'll see that nitrogen attached to carbon, and then you have this carbon double bonded to oxygen. So this right here is called a carboxylic acid group. This is called an amino group. So this is the basic structure of an amino acid. Now this R group right here can be 20 different things. So here you have all of the different side chains. So it's not important that you memorize all these, although you'll have the pleasure of doing that in a biochemistry course in college. Based on these different R groups, okay, so see here, we just have a hydrogen for R. Here we have, this is called a methyl group, okay, and they can be way more complex. They can have these ring structures. Um, they can have these long chains. But the basic part you're looking at is if you look, they all have the very exact same basic structure. Okay, so you're going to be recognizing amino acids because they have nitrogen. Carbohydrates did not have nitrogen. And we might just write that it's an R group, okay, or there might actually be molecules there, as you see in this chart of the 20 different amino acids. Okay, lipids are just like carbohydrates. They're made up of carbon, hydrogen, and oxygen. Okay, so you're going to have to learn to discern between lipids and carbohydrates. So I'm going to switch back actually to the carbohydrate slide. Okay, you'll notice that carbohydrates tend to be a ring structure. Okay, and then amino acids are easy to recognize because they contain nitrogen. Okay, and then lipids are not a ring structure. We're looking at this really long chain of hydrocarbons, and we say hydrocarbons meaning containing carbon and hydrogen. Sometimes we shorthand these molecules. Again, just like the ring structure of the carbohydrates, where we don't want to show every single carbon and hydrogen, we can just use a point. Okay, so you might see a lipid molecule drawn like this. This is this fatty acid chain. Each one of these points would be a carbon and hydrogen attached to it, but for short, we can just draw those lines. All right, and then we have saturated and unsaturated fatty acids. So you'll notice that in this molecule, we have all single bonds. Okay, carbon actually likes to make 4 bonds, which again, you'll learn more about in organic chemistry or even in your high school chemistry class. So if it has all single bonds, it's going to need to have lots of hydrogen. So it's saturated, it's covered in hydrogen. Whereas in our unsaturated fatty acids, we do have double bonds. It may just be one double bond like this one here. Okay, it could be 2, it could be 3. But either way, as long as it has any double bonds, it's going to be unsaturated. So we have less hydrogen in this molecule necessary because the carbon's double bonded to another carbon. So that's how you can kind of remember. So saturated, covered in hydrogen. Unsaturated will not have as many hydrogen. Or you can just simply remember that saturated fatty acids contain all single bonds, and unsaturated do have double bonds. All right, so again, this is just a reiteration of what I said on the last slide. So saturated are covered with hydrogen. They have no double bonds. They are solid at room temperature. So when you created your molecular puzzle pieces, you should have noticed that these molecules could easily stack on top of each other because they were in a straight line, which would give that uniformity to make them solid. These are typically considered our unhealthy fats, so like things like butter, okay, solid at room temperature, or the fats that are in animals. So lipids are going to contain these fatty acid chains. Here we have a glycerol molecule. Okay, and then we attach fatty acid chains to them. So unsaturated fats, you should have found that they have more room for more hydrogen, and they tended to have, because of this double bond, kind of a curve in their structure, which would make them less uniform and more likely to be a liquid at room temperature. These are healthier fats, so like if you've heard of omega-3 fatty acids or omega-6, That 3 and the 6 are as actually referring to where you would find the double bond. Okay, but basically, you really need to just know the difference between lipids and carbohydrates when you're given the structure. So again, long hydrocarbon chains is going to signal to you that we are dealing with a lipid, whereas a carbohydrate was a ring structure. Okay, and then we have our nucleic acids. So we talked about these earlier in the year. DNA, deoxyribonucleic acid versus ribonucleic acid for RNA. They're made up of nucleotides. So you should recognize these bases, okay, which kind of look similar to the carbohydrates, but keep in mind that these bases are going to be attached to a phosphate group and a sugar backbone. Okay, so let's talk about dehydration synthesis. So dehydration means to remove water, okay, or lacking water. Synthesis means to create. So we are going to create a molecule by removing water. So you guys did this in the lab. So you had your monosaccharide pieces, and in order to put them together, you had to remove oxygen and hydrogen from one of them, hydrogen from another, and then you were able to combine them to form your disaccharide. Okay, so in this case, we're removing water. So water would be a byproduct, meaning it's something that we create in the process, but it's not what we were trying to create. We were trying to create this disaccharide, right? And if we do this process over and over again, you're going to make polysaccharides. Okay. Hydrolysis is basically the opposite here. So hydro meaning water, lysis means to break apart. So we're breaking apart a polymer using water. Okay, so we're adding water to reform these molecules here. Okay, so we're adding back on this oxygen and hydrogen to this molecule and a hydrogen here. Okay, so that'd be 2 hydrogen, 1 oxygen, H2O, to recreate these monomers. Okay, hydrolysis often takes place in the digestive system, so we'll talk a lot more about this in the human body systems class. Basically, hydrolysis allows you to create energy. So hydrolysis of ATP is going to release energy. So when bonds are broken and new bonds are formed, energy is released. So there's a lot of energy in this phosphate bond here. So if we add water, it's going to break it apart, and it's going to produce, instead of adenosine triphosphate, now we have adenosine diphosphate. So one of the phosphate groups came off, and that's going to release energy. So again, we'll talk more about that in human body systems. Just really important you know now that ATP is energy in the body. So hydrolysis breaks polymers into monomers. Okay, so if we have a protein, that's going to break it up into amino acids. Carbohydrates or polysaccharides, starches, they're going to break up into monosaccharides or simple sugars. Now, technically, lipids are not polymers, but we can break apart the glycerol and the fatty acid chains. And then nucleic acids can be broken down into nucleotides. So if we have a whole bunch of nucleotides put together, that could make DNA, for example. We can break those apart into their monomers. And then using dehydration synthesis, you can do the opposite. So using dehydration synthesis, I could put amino acids together into proteins. Using dehydration synthesis, I can put monosaccharides together into a more complex carbohydrate like starch. Using dehydration synthesis, I can combine glycerol and fatty acid chains phospholipid molecules. Using dehydration synthesis, I can combine nucleotides into nucleic acids. So that's how we combine those molecules. All right. So building these bonds requires energy. So we would need to use ATP to create molecules. All right, so this is, again, was what I just went over on the other slide. So amino acids can be put together to make proteins, monosaccharides into polysaccharides. Glycerol can be combined with fatty acid chains to make lipids. So in this case, maybe if you have a glycerol and 3 fatty acid chains, that would make a triglyceride molecule. And then nucleotides can be put together to make DNA, so we can pass on our genes. Or these nucleotides can be put together to make RNA.